Thank you, Myra. So it's a pleasure to have Yosapol Anvanich today. Yosapol is a PhD candidate between two teams, so the team of Dolf Weijers and Joris Sprakel, so biologist and physicist. And so the topic is focused on the embryo development. So Dolf Weijers has made some seminal work on embryo development in plant science, and now it's turning to even more interdisciplinary fields, adding physics to the mix. And so Yosapol belongs to this new generation of interdisciplinarians, I would say. And so this is really what we want to encourage in the journal, actually, in the quantitative plant biology. And we believe this is, in a way, the future of plant science, this type of profile. So that's why we're very happy to have you, Yosapol. And the screen is yours.
Okay. Thank you, Oliver. Can you see my screen now, I think, right?
Yep, it works well.
Okay. Hello, everybody. My name is Yosapol Anvanich. I'm originally from Thailand, and now I'm a PhD student with Professor Dolf Weijers and Professor Joris Sprakel at Wageningen University and Research. And today I'm going to present our review paper that we published in Quantitative in Plant Biology about 2 months ago, in the topic of the Arabidopsis embryo as a quantifiable model for studying pattern formations. And here I'm going to show you why embryo is a good model and how to use embryo as a model for studying pattern formation. And the figure here show you Arabidopsis embryogenesis starting from very early stage to a late stage. And actually they have 3 main reasons to use embryo as a model for studying plant pattern formation. The first reason is that the embryo divide themselves in a highly predictable pattern. And this allows us to see the cell division process and also observe when, where, and how the cell are formed. And the second reason is that just only a few rounds of the cell division in Arabidopsis embryo, focus on Arabidopsis embryo, all the basic cell type in plants are ascorbate. In this case, is epidermal layers and ground tissue and vascular tissue. And this allows us to observe a cell specification process in a very minimal level. And lastly, we have plenty of genetic tools that are already available, for example, to observe a gene expression in specific cell or even observe in the subcellular structure. And this panel of marker lines show you all kind of subcellular structure that fluorescently labeled. And these all marker already available. And with these 3 main advantages, I think embryo is a very, very good model for studying a pattern formation and provide a very unique opportunities than the other models. And if we talk about a plant pattern formation process, it's always involved with 3 main steps. The first step is cell division. The second step is cell-to-cell communication. And the last step is cell specification. So we can start wherever. So I would like to start first with the cell division process. And I would like to explain that how to use embryo as a model for studying cell division process. In the past, the cell division in Arabidopsis embryo can only be observed in 2 dimensions. But now using DIC or light microscopy, but now with the current confocal technology, we can even observe in a 3D dimension. In addition, with a very powerful tool of a computational analysis of morphograph X programs or a PlantSec program, this is a kind of machine learning base. We can even quantify the embryos, the volume of the embryos and geometry of the embryo in a single individual cell level. And one of the example work has been done in our groups, previous colleague in our team. In this work, we quantify the embryos from a single cell stage to a 64 cell stage. We quantified the volume and we found that the volume of the cell decreased and less variant as the embryo developed. In addition, based on the observation, we do a simulation of Arabidopsis embryogenesis, comparing between wild type and mutants. And this allows us to better understand how the embryo of the mutant phenotype develop. And in this example work, show you how to use the embryo as a model to observe in 2D and 3D dimensions. But now with the development of the system, especially in vitro ovoculture system and live cell imaging developed by Kaitama groups, we can even observe the Arabiosid embryo in more detail. And here we can observe the cell division and cell specification process in real time. And this information allows us to map the developmental process of the embryos in a time series. And the same line of research developed by Minago groups. And in this work, they use Arabidopsis embryos with the live cell imaging techniques to observe the mitochondrial distribution since the first round of cell division in plant life. And this work was presented in the first regional club of this series. And to recap how to use embryo as a model for studying cell divisions, we can now observe the embryo in 2D, of course, and now in 3D, and now even in the live cell imaging with a lot of available cellular subcellular structure marker. And now I would like to move to the second components that are very important for pattern formation process as well, which is cell specification. In the past, several genes that involved in the cell specification process has been identified based on the conventional genetics. For example, ATML1 here expressed in outer layer and PL33 expressed for root stem cell. And this set of genes has been used as a marker to identify the specific cell type in embryo. But here you can clearly see that the number of genes is very that used for identifying the cell type in embryo is very robust and it's not saturated yet. And with the improvement of technology, we can understand deeper in the cell specification process based on transcriptomics data. And now we have a different data set of transcriptomic data starting from seed level to tissue, different tissue in the seed using the micro laser dissection. And now in the individual stage of the embryo, the transcriptomic data is also available. And in our groups, we generate a high resolution of transcriptomic data based on the specific cell type in embryo. And we generate the database called Albedo that's already available and very user-friendly. And in this database, it allows you to observe where and how the gene express in the different cell type and also in different stage of the embryos. But with the current available data, the gene expression cannot truly reflect the identity of the cell, because most of all the data here came from the pooling the sample. So the available data cannot represent the identity of those cells and also cannot reflect to their heterogeneity. And the most ideal transcriptomic data would be at the single cell level. And I think this would be the next step of the way to study the cell specification process. And one of the main challenge to start to generate the single cell data in plant embryo is to dissociate the cell. So, and this is the first step of the single cell method. And so far we can achieve using a mixture of enzyme combination and mechanical force, we can dissociate the embryonic cell into a single individual cell. And this is what we achieve so far. And to recap, in cell specification process, we have identified 0 genes involved in the cell specifications, and we have plenty of available transcriptomic data starting from seed to cell and even single cell type. And the next step will be at the single cell level. And now it's about the cell communication, which is a very important process as well for pattern formations. And here I would like to focus on the plant phytohormone auxin. And most of the pattern formation process in plants, I would say, depend on the auxin biosynthesis and auxin transportation and auxin accumulation as well. And the mutant of the auxin biosynthesis and auxin transportation lead to a very severe defect in the embryo patterning. And so far is several of auxin biosensor has been developed, and I would say that what we see here is kind of the outcomes of the mutant. Is what we see in the phenotype. The outcome is an output of the process, but it is very crucial that we need to visualize auxin and quantify auxin properly. And several auxin biosensor has been developed. And one of the most widely used is DR5 reporter line. And with this reporter line, we can be able to visualize auxin and see accumulation, see auxin accumulation. And but the problem here is that the auxin here can only reflect in the maximal level, and we cannot detect the auxin change or auxin response using this reporter line. And in our groups, we develop more sensitive reporter lines, call it R2D2. And with this reporter line, it allows us to detect auxin response and auxin change over time, and be able to quantify the auxin at individual cell level. And beside auxin, other phytohormones and other micromolecules, for example, mRNA, also play a very important role in Arabidopsis embryogenesis, but we still lack of two, and there's still plenty of room to develop the two and to be able to study in the other phytohormone and molecules. So it's still plenty of room to develop. And to recap in these sections, cell-to-cell communications, we can visualize auxin and quantify auxin at a single cell level based on the available reporter line. But beside auxin, other phytohormones and molecules also play important role, but the two still need to be developed. I think now I cover three main important processes that involve in plant pattern formation, which is cell specification, cell division, and cell-to-cell communication. And if I conclude this process in the very simple word, I would say it's as a biochemistry process. But in fact, not the biochemistry process is involved in the pattern formation, but also in physics. And if you think about the cell wall that connect together, so we have some force or some mechanics between cells that involve in the pattern formation. But in plant, it's very difficult to study those mechanical properties, and it is very difficult to study physics in plant. And one of the most widely used approach is detect or quantify the cellular mechanics based on atomic force microscopy. But one of the problem in using this approach is that we can only detect the mechanical force of outer layers of the surface of the cell, but we cannot quantify or understand the inner layer or the mechanical property of the inner layer tissue. And in Sparkle Group, we developed the probe that we can quantify the cell in the inner tissue. And basically, we develop a fluorescent dye that can attach to a specific location of the cell or cell wall or plasma membrane and be able to quantify the mechanical property of those surrounding. And the fluorescent light here reflect to the mechanical properties of surrounding the cell. And this is what it is already available, and we already test in stomata and roots, but not in embryo yet. So now we have an idea how to use embryo to study the biochemistry process, and we have an idea how can we study in the aspect of physics. But the next question is how to link between the physics and the biochemistry. And actually, this is my current research. And what I'm doing now, I have a question that how the plant feel the force. Yeah? And what I did, I designed a microfluidic device that can precisely apply the force to the embryo. And I do a simulation and also the segmentation to make sure that all the cell receive the force from this squeezing. Yeah, and then I'm trying to study the gene expressions and the pathway that involve to this mechanical stimulation. And I hope that from this information, we will understand better how the plant feel the force and how the plant respond to a mechanical cues. And to end up with these presentations, I think we know quite so far in biochemistry regarding the pattern formation in embryo, and we start to understand better in terms of physics. And I think the next step or the future direction of this research career would be the combination of biochemistry and physics. In the end, I would like to thank my supervisor, Professor Dole Byers and Professor Joris Sparkle, and also Dr. Vera for all guidance or comments and very great support. And I would like to thank Lucy as well and Dr. Joao for feedback. And actually, Lucy is the first author of the mechanical probe sensor. And lastly, I would like to thank the resource of grants that I'm currently doing on the research, NWO and ERC. And thank you very much for your attention. And I'm very happy to answer your questions. Thank you very much.
Thank you, Josapol. It was super clear, very clear. Thank you very much, and a very cool, cool tool. Maybe I can start with a question. I see that there's already some questions, so maybe I should give the priority to people asking questions. So, question from Christopher Skilbeck: what role do you see for network analysis in understanding pattern formation in the embryo?
We the network. Actually, we didn't get those transcriptomic data yet. We are now on the process of generating, but of course, I mean, we applying we're applying quite a critical force to the embryo in a very short time. So I expect to see some change or some fast response. Yeah, it's a bit difficult to guess what is there, but I think, for example, some expansion or some genes that involve like DEC1, this kind of thing would be expected to be observed, but yeah, it's a bit hard to predict what the network analysis would be or what the interaction would look like.
Okay, and even independent of the mechanical stimulation, so in terms of computational modeling, do you plan to do, or your group plan to do network analysis of, I think maybe Christopher was also thinking of interactomes, or more like a really network in the larger sense, right? So how do you say gene network analysis, or
yeah, that's what we plan to do as well. But I think it's to be able to construct a network analysis, I think in my opinion, I think we should be able to see what the gene is actually respond, and then we link to that to see what the pathway involved and what is the negative feedback on that. But yeah, to starting from the network analysis, I think yeah, in my opinion, I think it might be a bit difficult to dig deeper in what the biological meaning on that. Yeah.
Okay. Yeah. Fair enough. It's good to have an answer. So there's another question from Thomas Stanislas from Tübingen.
Yeah.
Very interesting talk. Quick question: in your microfluidic device, you can only apply a mechanical stress on one direction, whereas in vivo mechanical stress occurs in all directions. Do you think it will be
true.
a problem?
Yeah. That's it will be a problem, and I don't think it will be a problem, because I think that the idea of the research that we apply the microfluidics is that we don't interest much about the direction of the force at the moment, but we are more focused in the general basis how the plant feel the force. So if they apply in the different directions, but the cells still feel the force, I think that's okay at the moment. And what we use microfluidics, because I think it's only available tools. That's what I can think, that can precisely apply the force to the embryo at the moment. Yeah. And with the simulation that we do and segmentation, we kind of we make sure that all the cells perceive the force based on the volume change that we observe. So we stimulate. We do the simulation before squeezing and after squeezing with the same embryo. So that's my idea. But yeah, it's a very good comments on this. Yeah.
Maybe actually, okay, it's a follow-up question from Thomas. And the embryo is still dividing in the device. Is it still dividing?
No, it's not dividing. So we extract the embryos we control the state of the embryos to make sure that it's not have a lot of variation between the developmental process. So we focus on particular state of the embryos. We extract it and squeeze it, and then extract RNA immediately. Yeah. Okay. So we wait for we wait a bit for mechanical response, and then we extract the embryo immediately. So it's not dividing. Yeah. It's not. Yeah.
Okay. Yeah. And actually, I had a question also on this. So do you have an idea of the exact stress pattern when you do this? Because it's probably. I mean, locally you have probably some friction. In some case, you compress some tissues, but you put tension here. So it's likely a complex stress pattern, right?
Yeah.
And yeah. So you do this with morphodynamics, I guess, or how do you calculate the stress pattern in this? I mean, in the
yeah. So we plan to do in the morphodynamics. Yeah, that's a point. So what we think is that I think we already have available, for example, microtubule line, and by the squeezing, and then we observe under the squeezing positions. I think we should be able to stimulate or do the simulation based on a stress pattern. But in this, as you said, I mean, it's quite a complex stress, maybe from the shear force or not only mechanical stimulation, but we're trying to minimize those forces as well by coating the surface. But this is as much as what we can. Yeah.
No, but it's actually nice to have a complex stress pattern, because if you can do a single cell analysis, and it could be super interesting. There's another question from Thomas. Extracting RNA means that you are looking for long-term response and not immediate response, protein relocalization, for example, to mechanical stress. it's my last question.
Yeah, actually, yeah, that's right. But I think it's depend on how much the time frame you put on that. But I think for in my case, I extract RNA. After the embryo pass to the channel, I wait for 30 minutes to make sure that they have some response, and then we extract RNA. That might be a bit still long, but at least we would like to the point is that we don't know how the plant respond to the force, and we don't know how fast they respond. So we kind of trying to play with the spectrum that we should be able to see something, and we start to adjust based on the threshold of mechanical stimuli. Maybe they have a very fast response that, okay, when the embryo out of the channel, we can immediately extract and they see the gene expression. But the point is that is no evidence on that, and then we don't have a knowledge on that. So in my project, this is a starting point that explore, let's say. Yeah.
Yeah, of course. Yeah. No, there's plenty to do. I mean, I was actually thinking as well as maybe a follow-up for the future is to do oscillating constraint, right? So if you change the flux in the microfluidics, you could have a sort of frequency in the stress, and then see. I mean, there's some people who've looked at this and see that there's some acclimatization to the stress, right? So if you repeat the stress, the cell is sensing it less and less. So it could be the same in the embryo. Actually.
You mean during the squeezing?
During the squeezing, yeah. So
squeeze. Okay. Okay. Oh, that's a very interesting point as well. Yeah. Yeah.
Probably
But
more
I'm not sure that we have a current evidence, or maybe you can suggest as well. Do we have any idea how fast the mechanical stimuli respond in plants? I mean,
for this acclimation, it would take. I mean, the only study I know is from trees, so it takes several days. Huh? So this is really much more long term. But there might be some more short term effect like this within minutes. Or
That's
follow-up.
Follow-up,
Definitely more long term. I was also, so when you were showing the patterning with hormones, which of course, I mean, auxin is super important, but I'm always thinking, I mean, there are many other signaling molecules, right? And I mean, one of the, I mean, maybe the most abundant one is peptides, actually. So is there, I mean, on the embryo, actually, I'm not so aware of the picture for peptides patterning in the embryo. I mean, do you have an idea on this, or?
So far, in my experience, I would say I didn't see that much. At least in the studying in the embryos, I didn't see that much. But what I see, for example, some hormones, cytokinins, and micromolecules, mRNA, I see some study. Yeah, but mostly based on the in H2 hybridization, but not in the 2 as a sensor. And for protein, I in my experience, I didn't see.
Okay. No, but yeah, I mean, there's plenty to investigate. So maybe I can ask one or two, or maybe I should, maybe one, one before last question, maybe. I'm convinced that the embryo is a great system. I'm also thinking that's maybe the question I already asked Dolph a few years back. So when you look at embryo, there's also the somatic embryogenesis. So you can have embryo outside. So of course, the development is very different. But is this something that could be of interest? I mean, I'm thinking in terms of accessibility, of course, it's much easier. But is this something you're thinking about? Or
yeah, they have a research that zygotics and fertilized embryo is not that different in the gene expression. But I think in terms of culture, at least in my opinion, I would prefer a normal embryo, fertilized embryo, and extract from Arabidopsis. I think we have quite a good approach that already proposed as well, not by our groups, but we follow that protocol as well. And I think that that should be feasible enough. And we have the protocol that we can do RNA extraction, and this protocol developed by Micronodins. To extract RNA in a few number of embryos. So in fact, I think in terms of replication, if you grow 1 tray or a 2 tray, I think it should be feasible instead of trying to optimize in the culture system. And in terms of patterning, I never observed in embryo cultures. But I believe that the patterning should have some at least minor difference, because if you think about in the Y type embryos, they will have endosperm outside. That is control mechanical patterning. So I think that do directly from the plants would be the easiest way. Yeah.
Yeah, for sure. Yeah, we got. I understand. Okay, maybe one really last question. It's more like an opening. Let's say so. Since you have this profile between physics and biology, so maybe I guess 2 questions. So, why, in your case, did you move to this field, like to this interdisciplinary question? And maybe the second question would be, what would be your advice to someone, a student or anyone, who would want to, a biologist who would want to do some physics, or a physicist who would want to do biology? What's your experience and the feedback you can share, right? With the community?
I think the world of science is so big, right? And what I see is that you can use biological tools to answer the question in biology, but also you can, if you opens a bit on the knowledge in, for example, in physics or in engineer or in mechanics, is we make you think in a very different way, and the way to answer the biological questions would be more complex. But I think to conclude in a simple word, I think with the combination of biology and physics, we can understand science better. And I think that's a main point. And for your second question, I think that the world of science now is very multidisciplinary, and one person as a biologist can be a bit of physics as well, and a bit on the computational analysis as well. And I think mix between the different things can lead the science more exciting. That's what I feel. Yeah. So I'm very encouraged to people in different field to jump across the field as well.
Sounds good. I think that's a nice conclusion from your talk. So thank you again for a very, very clear presentation and very inspiring. So thank you, Yoza Paul.
Thank you very much. Yeah.
I'll hand over.